Good evening, dear students. In yesterday's class, we discussed about the preparation, types of second type of amino acids and preparation. We discussed the two preparations. First one is by elimination alpha halo acids. Second one is a Straker synthesis. And third method, I am discussing third method in today's section. The preparation of amino acid. Heading is preparations of amino acids or methods of preparation. Methods of preparation. The third one, bi-Gabriel synthesis. Bi-Gabriel synthesis. Okay, this is a third method. Here, an ester of alpha halo acid is treated with potassium thalamide to form the corresponding substituted thalamide, which on hydrolysis gives phthalic acid and an amino acid. Here, an ester is treated. The ester of alpha halo acid, the presence of example ethyl chloroacetate. Here is in ethyl chloroacetate, and it is treated with potassium thalamide to form the corresponding substituted thalamide, which on hydrolysis gives phthalic acid and amino acid. Here I take first the potassium thalamide. This is a potassium thalamide. Potassium and potassium present, potassium thalamide, sodium present, sodium thalamide. Here this is a potassium thalamide. This is on treating it, this is a negative charge and this is a positive charge, and it is treated with ethyl chloroacetic acetate. Ethyl chloroacetate ClCH2 plus is treated with ethyl chloroacetate ClCH2 COCH2 CH2. Then it is removal of this KCl, and this is attached to this one. Here the corresponding substituted thalamide is formed. Substituted thalamide. This is substituted thalamide. This attached here CH2 CO CH2 CH3. Or you can write here C2H5. This is also your C2H5. Okay, here removal of KCl. This one is KCl. Substituted thalamide is formed here. Write this one. Substituted thalamide C double bond O, C double bond O, and presence of N here. Here this is substituted CH2, COO, CH2, CH. This is a substituted element. Okay, then it is hydrolysis takes place. Reheating hydrolysis takes place. Here, this is a continuation reaction. Here, hydrolysis takes place. I will write the continuation reaction step. I will write here. The formation is phthalic acid and amino acid. Sorry, yes, amino acid, phthalic acid and amino acid and ethyl alcohol. On hydrolysis of that substituted thymine, this get the phthalic acid. COOH, carboxylic acid group is present. Here, this COH, this is a phthalic acid. Phthalic acid plus amino acid is formed here. Plus NH2, CH2, COH. This is a amino acid. Amino acid plus another byproduct here plus CH3, CH2, OH. On hydrolysis, the ethyl alcohol is formed. CH3, CH2, OH. This is a Gabriel synthesis. It forms amino acid. Here you are preparing the methods of preparation of amino acids. Amino acid, it form a amino acid. This name is a glycine. The special name is glycine. This is a glycine. The amino acid is formed here. First is the potassium thiamine is treated with alpha halo acids. This is a fluoroacetate, ethyl fluoroacetate. This is the ethyl group, ethyl fluoroacetate, and substituted thiamine is formed. Then this is hydrolysis takes place here. Then form the phthalic acid and amino acid plus that is ethyl alcohol. This is very important for 4 marks question or 3 marks question. In examination, write the compound name here. These are the methods of preparation of amino acids. Next, Zwitter ion. Sorry. Next one, Zwitter ion. Here, what is meant by Zwitter ion? This is very important for 1 mark question. Define Zwitter ion or what is meant by Zwitter ion. 1 mark question is very important for this Zwitter ion. Zwitter ion is defined as a Zwitter ion is an amino acid molecule containing a both positive and negative charge. Here, the amino acid molecule containing both negative and positive charge. This is called as Zwitter ion. See here, Zwitter ion means NH2, CH2, COH. Okay. This is when dissolved in water molecule. This is dissolved in water molecule. Then here it loses the carboxyl group loses proton and NH2 group accepts proton. This is accepts. This is loses. And it get a positive charge and it get a negative charge. It presents so positive as well as negative charge in amino acid that is known as the bitter. Right here, the NH3, CH2, COO minus. This get a negative charge. This get a positive charge. This is known as Zwitter ion. Okay, here I explain this Zwitter ion. You know what is meant by Zwitter ion. In exam or screen examination, the 1 mark question, you write a particular definition. A Zwitter ion is, I write here the definition of Zwitter ion. A Zwitter ion is an amino acid. A Zwitter ion is an amino acid molecule containing both amino acid molecule containing both positive and negative, both positive and negative charge This is called as Zwitter charge. It is called as Zwitter ion. You write in examination this definition define when asked in examination for 1 mark question define Zwitter ion. A Zwitter ion is a amino acid molecule containing both positive and negative charge. It is called as a Zwitter ion. Okay, here Zwitter ion, it is a amino acids are a bifunctional group. This Zwitter ion is a bifunctional group or bifunctional compounds. They contain both acidic group, that is a COH group, and basic group NH2 group. The both acidic and basic group. Here this is a carboxyl group and this is a amino group. These two are contained in a amino acid compound, and it is a bifunctional compound. And here amino acids undergo ionization in water. When amino acid under ionization in water, amino group accepts protons and sewage the carboxyl group. Here it loses the proton. Here the carboxyl group loses the proton, and whereas amino group accepts the proton, whereas amino group accepts the proton, the result of this zwitter ion. Result is zwitter ion. The formation of zwitter ion will be formed, which is electrically neutral. The zwitter ion is electrically neutral. Here, the formation because of the presence of both the acidic and basic. Because of this is a electrically neutral. Here, the losses of this carboxyl group. Here, presence of here. This is a carbon. This carboxyl group here, carbon double bond O, and here OH. And formation the when ionizes deprotonation, ionizes deprotonation or loses proton. See here, what we write here? Carbon double bond O, and this is O. This get negative charge plus H. Here losses of hydrogen. This is losses of proton. This is a reaction, and dissolving or ionizes or deprotonation or loses the proton. This is a from amino acid. From amino acid. This is a from amino acid. When this amino group accepts protons here, write this reaction. Okay, this is a N and two hydrogen here. This here presence of lone pair of electron. This is hydrogen, and this is a hydrogen plus proton is accepted here, and formation is protonation. Here accepts proton. In this deprotonation takes place. In this protonation takes place. Protonation or accepts proton. Here formation is N is positive charge here, H, N, positive H and H. Get a positive charge. Finally, I will write here. I take an example for amino acid. For this zwitter ion, I have already given the example for zwitter ion. Amino acid, I have taken glycine. Here, glycine NH2 CH NH2 CH. When it is protonation takes, or can write here substituted H and here ionization. Here ionization takes place or in water. It is dissolved in water. It gets H2 is a NH3 and CO is loses the proton. Here I write in water its formation NH3 CHCO minus. This is known as Zwitter ion. Okay, here presence of positive charge and presence of negative charge. You told here this is a Zwitter ion contains both positive and negative charge. Here positive charge and here negative charge. Zwitter ion, it is also known as a dipolar ion or this dipolar ion. Dipolar ion. In examination, define dipolar ion, you will define the Zwitter ion. You write Zwitter ion definition. Okay, here the neutral form of the Zwitter ion is a neutral, electrically neutral. The neutral form of amino acid, here the neutral form of amino acid is known as Zwitter ion. And it is electrically neutral and therefore the amino acids are considered as amphoteric acid. So amphoteric and therefore the amino acid act as both acid and base. Here the amino acid act as both acid and base. Here the presence, because the presence of positive and negative charges, and it is considered amphoteric. And this the neutral form of amino acid changes its form depending upon the pH of the medium. Here the neutral form of amino acid changes its form depending upon the pH of the medium in which the amino acids are dissolved. They change to acidic form or basic form depending upon the medium. Here alright, the reactions, it is depending upon it is acidic medium or basic medium, it changes to a V. Alright, here the reaction. Just depending upon the medium, the reaction changes here. This is a CHRNH3 positive OO negative. This is a Zwitter ion. Sorry, I lost here. CO. When COH is present, the proton is losses only. The CO minus is removed. This is a Zwitter ion. This is Zwitter ion or neutral form of amino acid or neutral form of amino acid. Neutral form of amino acid. Here, when this amino acid, this neutral form of amino acid or Zwitter ion is dissolved in sodium hydroxide, the basic medium, sodium hydroxide, NaOH and H plus. Here, this is a basic medium. Here removal of water molecule. This jitter ion is treated with sodium hydroxide, basic medium, removal of water molecule. It formed here the NH2 is formed, H, R and CO minus. Negatively charged anion is formed. Negatively charged anion means is a acidic form migrate towards anode. The acidic form migrate towards anode. The formation is here the same NH2 CHR minus. Okay, this name is the negatively charged anion. Negatively charged anion. In this side, you treat with acidic, it forms a positively charged cation. It forms a negatively charged anion and the acidic form migrate towards anode. Acidic form migrates towards anode. This is similar to here, only changes positively charged cation and basic form migrates towards cathode. Basic form migrates towards cathode. Once again, sorry. Here NH3, this is a jitter ion, this is a sodium hydroxide and basic medium. This sodium is a base, this is a strong base and here it is basic medium. The removal of water molecule form here this H and this H. Here the water molecule is removed. The NH2 and CO negative form is formed. The anion is formed. Negative charged anion is formed. When it get negatively charged anion, it acidic form. At negative charge, the acidic form migrates towards anode. Here they treated it HCl, acidic medium. Here treating the acidic medium. I am writing the animal this side. Okay, HCl, H plus. This is a acidic medium. This is a acidic medium. Acidic medium pH. Okay. Here the formation is NH3CHCOOH. Here removal of this alkyl group here. The formation is here the presence of NH3HRCO minus and the presence of H and here get a positive charge. It accepts the proton. Here the formation is NH3C and here HRCOOH. This is a positive charge. It get a positive charge. And here the positively charged ion. Here positively charged. Here in that side no positive charge, presence of only negative charge. Here the positively charged ion. This is a positively charged ion and positively charged cation. Here in that side it written anion. Here cation, positively charged cation. Here the cation basic form cation basic form, cation basic form migrates towards cathode. Migrates migrates towards cathode. Migrates towards cathode. Okay, this is a reaction depending upon the medium. This is a reaction for Jupiter ion, and it depending upon the medium. And neutral form of amino acid changes to positive charged basic form, positive charged basic form, and in presence of acidic solution. Positive charged basic. Here the positive charged basic form in presence of acidic solution, whereas it changes to negatively charged acidic form in presence of basic solution. And this ionized amino acid solution, when it is kept in electric field, basic form migrates. When it is kept in electric field, the basic form migrates towards cathode. This basic form migrates. This is a basic form. It migrates towards cathode. Yeah, and acidic form of amino acid migrates towards anode when kept in electric field, and neutral form zwitter ion will not migrate. Here this zwitter ion is will not migrate towards any electrode, because here this is a neutral. There is here it is a electrically neutral. It does not migrate any electrode. This is an explanation of zwitter ion. This is only one mass question. Only define zwitter ion or water zwitter ion. Next point, next concept, isoelectric point. Next one is isoelectric point. Isoelectric point. Okay, here this is also important for your examination. For your exam, yeah, in exam asked for 1 marks question. This is important for 1 marks. Isoelectric point. Here the isoelectric point, definition of define isoelectric point or what are isoelectric point. Here definition of isoelectric point. The isoelectric point is the pH at which an amino acid exists completely as the zwitter ion. This is known as a isoelectric point. The pH at which amino acid do not show any tendency to migrate when placed in electric field is known as isoelectric point. You write this definition of this definition. I write two definitions here. The isoelectric point is the pH, the isoelectric point is the pH at which an amino acid exists. An amino acid exists. An amino acid exists completely as the zwitter ion. Completely as the zwitter ion is called or is known as isoelectric point. Isoelectric point. This is a definition. Isoelectric point. The isoelectric point is the pH at which an amino acid exists completely as the zwitter ion is known as isoelectric point. Or same here, the meaning is same here. The isoelectric point, the pH at which amino acid, the pH at which amino acid, do not show any tendency to migrate when placed in electric field. To migrate when placed in electric field. Is known as isoelectric point. The isoelectric point. The pH at which amino acid do not show any tendency to migrate when placed in electric field. That point is known as the isoelectric point. You write this one in your examination. Okay, here the isoelectric point explanation here. The only explanation of isoelectric point. This is only definition is very important for your examination for 1 mark question. Okay, here the isoelectric point of amino acid is characteristic property. This isoelectric point of amino acid is a characteristic property, and each amino acid will have its own isoelectric point. Each amino acid have each isoelectric points, and it is constant like melting points, and it is constant like melting points and other boiling points. And here therefore it can be used to identify the amino acid. It is the isoelectric point. It is used for the identifying the amino acids. And I will write here some isoelectric point of amino acids. The isoelectric point of glycine is the pH is 6.1. pH 6.1. Alanine, here the pH is 6.02. Next align is 5.97. 5.97 and serine is 5.70. 5.70. These are the pH of this isoelectric points here. The glycine is a 6.1, alanine is a 6.02, and valine is a 5.91, and serine is a 5.70. Okay, stop here class. In next class, I will discuss the remaining part of this amino acids. That is a electrophoresis. Thank you.